ma'am, all have come join, ma'am. It's a YouTube link, ma'am. Yes.
Okay, I can't see you all at the moment, but I guess after my talk I'll be able to see you. So I'm just imagining you all sitting there. I hope you had a nice lunch. Good
Yes, ma'am.
afternoon. And
Yes, ma'am.
I'm very happy to be with you again after 2 years
Yes, ma'am.
and 2 difficult years, right, for all of us. So, shall I start my
Ma'am,
talk now?
we shall start with the prayer song, ma'am, or five
All
minutes,
right.
ma'am. Kindly wait. Yes.
Sure.
I'll introduce you, ma'am. Then you shall start, ma'am.
Okay, great.
Yes, yes.
This is the day the Lord has given to us. Let us rejoice. I welcome all of you to this international webinar through this online platform. Prayer is the
Yes.
song of heart raised from soul. So let us invoke God's presence with this prayer song.
As we gather in this place today, Holy Spirit, come and have your way, have your way. As we lay aside our own desires, sweep across our hearts with fire, have your way. This is your house, your home. We welcome you, Lord, we welcome you. This is your house, your home. We welcome you today. Lord, we welcome you today. And suddenly you just know it's time to start something new and trust the magic of beginnings. So I request Dr. Carolyn Jo Rosario, Head Department of Botany, to welcome this new beginning.
Give thanks to the Lord. Give thanks to the Lord for his good. His love endures forever. Psalm chapter 107, verse 1. Good afternoon to everyone gathered here. I take this opportunity to welcome you all for the international webinar organized by the Department of Botany, the Bimla College for Women, Coimbatore, on the theme RNA for Biotechnology. I invite to welcome our resource person, Jennifer Ann Harikrishna, Dr. Jennifer Ann Harikrishna, Genetics and Molecular Biology, Institute of Biological Sciences, Faculty of Science, University of Malaya, Kuala Lumpur, Malaysia. She completed her doctorate in molecular genetics in Cranfield University Microbiology, University of Surrey, UK in the year 1985. Her professional experience includes she is the director of the Research for Center in Biotechnology for Agriculture and professor of genetics, Institute of Biological Sciences, Faculty of Science, University of Malaya from the 2011 till present date. And she has a H index of 15 in the Google and 12 WOS. And she is the author of 65 peer-reviewed scientific publications and over 1,000 citations in Google. And widely, she has been sought as an examiner, editor, reviewer, and invited speaker at international scientific meetings. And from the year 1983, her career started up till the till date. And she has received Whiteboard Scholar Award for the PhD studies from the year 1985 to 1989 and awarded the Cranfield University Chancellor's Gold Medal for the most outstanding graduate student for the year 1990 for industrial research leading to UK patents for the brewing industry. Her leadership and management skills includes she has been the director of CEBAR UM from the year 2011 to present date and supervising a group of 12 academics, 6 support staff and 20 graduate students. She is leading 9 projects valued approximately 5 million and managing a center annual budget of 1 million covering 6 laboratories and supporting facilities. And she has published more than 70 articles in the reputed journals and 7 book chapters. She has been selected as invited lectures in several international scientific meetings and as keynote speaker in conferences. And he has been as a consultation from the year 2015 to till date, from the year 2000 to till date. And she has received many awards like recipient of Excellent Science Certificate in the year 2018, University of Malaya Research Invention and Innovation Expo in the year 2010, and she has been got gold medal for the year 2009 under University of Malaya Research Invention Innovation Expo, and Gold Award in the BioInno Awards in Bio Malaysia from the year 2008, and Gold Award International Invention, Innovation and Technology Exhibition in the year 2008, and Best Poster Award for the 32nd Annual Conference of the Malayan Society for Biochemistry and Molecular Biology, and Bronze Award and the Lord King Staunton Gold Medal Award from the Cranfield University in the 1990, and so on. She has got patent for 13 files from IPR, and her teaching and supervision, and she has completed 11 PhD scholars and completed masters 15 and 19 and a mixed board, and completed many MSc projects also, and she has at present she's guiding 7 PhD scholars in the ongoing University of Malaya. And her evaluation activities include she is the reviewer for scientific journals such as Animals of Botany, Aquatic Botany, BMC Plant Biology, Frontiers in Plant Science, Journal of Experimental Botany, Journal of Agrobiotechnology, Malaysian Applied Biology, Malaysian Journal of Science, Molecular Journal Biology Reports, and Plant Growth Regulator Journal, Tree Genetics and Genomics, Tropical Life Science Research, etc. She has been the internal examiner for the many universities, especially University of Malaya, an external examiner for PG, an internal examiner for MPhil, MSc, and MSc Biotech, and PhD. And she has been external examiner for MSc in the University of Cape Town, South Africa, University Putra, Malaysia, University of Malaya, Sabah, AIMST, University of Kemsagan, Malaysia. And she has been the external examiner for PhD in universities like Murdoch University, Australia, VIT University, Vellore, University of Nottingham, Malaysia, University of Kebasgan, Malaysia, Monash University, Malaysia University of Science and Technology, Taylor's University, Malaysia. She has been the external reviewer for academic staff promotion in the Taylor's University from the 2017 to 18, and external evaluator for promotions of academic staff at UCSI University near 2017, and external evaluator for promotions of academic staff from the 2017, 15, and 12 in various academic institutions. So we are happy to have such eminent and intellectual person. We are enormously happy to have you, ma'am, in spite of your busy schedule. On behalf of our management, with great pleasure, I welcome you, ma'am. I also welcome our respected secretary, Reverend Dr. Sister Ruby Alangara Mary. I extend a warm welcome to our dynamic principal, Reverend Dr. Sister Helen, for her constant support and encouragement to organize this seminar. A hearty welcome to our beloved vice principal, Reverend Dr. Sister Mary Fibiola. I also welcome our controller of examinations, Reverend Dr. Sister Emelda Mary, and our Dean of Student Affairs, Dr. U. Mary, and Dean of Academics, Mrs. Valencia Kamal, and IQAC Coordinator, Paulina Santi Joseph. A warm welcome to all the faculty members from our department, research scholars, and students of our department. Once again, I welcome you all. Thank you.
Thank you, ma'am. As the deer searches for water, we search for wisdom. So I would like to invite Professor Dr. Jennifer Ann Harikrishna to enlighten us. Please take over the session, ma'am.
Thank you very much, especially to Dr. Carolyn, but to everyone for the kind invitation, and to all of you for being here with me this afternoon. It's always a little bit more difficult when we are online, but I hope you will not feel shy to ask me questions, and we can interact after the seminar. If the slides do stop changing, do let me know, because I can only see my screen. So I hope everything will run smoothly. And again, I'm really happy to be with you again after 2 years since I last joined you. So today I want to talk about RNAi, or which is short for RNA interference, and its use in plant biotechnology. And the examples I'm going to give you relate to orchids. That's why you see so many pretty flowers on the slide. And they truly are one of God's great creations. So as you were saying your prayers, I think we can always appreciate the beauty of his nature and in the flowers. And however hard and clever we might be, it's still a lot of mystery to us about how genetics works, and how to control it, maybe to improve or change these organisms. So I'm going to start with a little bit of background about University Malaya. We have an MOU together with you, and we're trying hard to work together. It hasn't been easy because of these past 2 years not allowing people to travel, but hopefully in the future, some of you may come and visit us, and I hope I will have the chance to come there and visit you in person. I'm then going to give introduction to the topic of RNA silencing, RNA interference, and why we're using that technique in our studies. And in particular, we are using it for orchids, although we are using it on other crops as well. But today I should just give you the examples from orchids, and we're using it for plant protection and for novel flowers. So University Malaya is in the peninsula of Malaya, which is part of the country of Malaysia. And the university is older than the country. That's why it's named Malaya and not University Malaysia. Later on, after the formation of the university, some states on the north of Borneo Island became part of the Federated States of Malaysia. And so our name reflects our geographical location. And the main campus is in Kuala Lumpur. The original campus was actually in Singapore because Singapore was briefly a part of Malaysia for about 2 years. Our center is for research in biotechnology for agriculture. It was established in 2005, and it's one of 6 centers of excellence of research in University of Malaya. We have many centers, but these 6 particularly focus on research. We have 6 labs, and we have facilities for doing contained growth of genetically modified plants and for testing them in greenhouse environment. We also have tissue culture facilities and bioinformatic facilities, as well as our molecular labs. We're also very proud of our ranking. In fact, this year it was just announced we have moved up to number 8 in Asia in the QS rank. So we are very proud of that position. And it means that we do have a lot of colleagues who are very good in their fields, which is really helpful to us when we want advice from different centers. So the molecule we're going to be talking about is RNA. And RNA is a very important form of nucleic acid. Of course, we know DNA is very important. It has all the information that the organism needs to grow and develop. But the DNA stays as a sort of memory bank, and it is the RNA that is the active molecule. It is essential for the coding, decoding, regulation, and expression of the genes. So it is copied or transcribed from the DNA into this message, and it is then made into proteins using ribosomes. The ribosomes themselves also have RNA components, and the transfer RNAs bring the polypeptide chain. So we can see RNA is involved in all sorts of stages like this. RNA itself is actually regulated by RNA molecules as well as by proteins. So not all RNAs are just encoding, they are actually active molecules just as they are part of ribosomes, but other parts of regulation. And RNA interference, which also sometimes is called RNA silencing, and you may hear me sometimes say RNA silencing instead of RNA interference, but they mean the same thing. It's a process that can be used to stop one or more proteins from being made. And this is actually a natural mechanism that occurs in all organisms, from bacteria up to mammalian cells, as well as plants. So the first thing, although we're going to use it in biotechnology, is to say that it is a naturally occurring process. And in this process, RNA molecules inhibit the transcription or the translation of the gene. And we can separate it into these 2 processes. When it's inhibiting the transcription process, we'll call it transcriptional gene silencing. And I'm not going to be talking about that form today. When it inhibits the translation, we call it post-transcription. That means that the RNA is made, but we interfere with it being used to make proteins. And this can be referred to as post-transcriptional gene silencing. And it's the most common form when we're talking about RNAi in most contexts, especially in biotechnology. We're talking about the post-transcriptional level. So RNAi is very commonly used for gene silencing in humans or animals. In plants, we also call it RNA silencing, RNA interference, and the form I'm talking about today, the post-transcriptional gene silencing. So, as we mentioned, it's a natural process. It's going on all the time in plants, and it was observed long before it was understood. So I took this from a review by David Bolcom, who is one of the foremost researchers in the field of RNA biology in plants. And he mentioned in this review paper in 2004, a paper that was published back in 1928. And this was by a researcher called Wingard, who was looking at virus infection in tobacco plants. And you can see the leaves down here are spotty and deformed. They're infected with the virus, and they clearly show the symptoms. But as the plant grew and developed, he noticed that the new leaves were free of virus symptoms. Now, if he rubbed these new leaves with the old leaves to infect them with the virus, they still remained without symptoms. So it looks like the plant has recovered from the virus. So they're asymptomatic, resistant to secondary infection. And this recovery wasn't understood back in 1928, but it didn't stop people using mild strains of viruses from protecting plants against more severe strains. So it was empirical evidence that something was happening, but they didn't know how the plant was able to protect itself. What this first study shows us is that viruses are great targets for RNA interference. Although they didn't know it was RNA interference, it was actually RNA interference that was helping this plant to protect itself. If a plant can naturally protect itself, then we can use this in biotechnology to protect plants from viruses. I'm going to jump forward a long way into 1990 when people were first doing genetic engineering experiments in plants. So this was the first work where people started adding genes into plants. So GM technology. This group wanted to alter flower color. They took chocon synthase, which is one of the enzymes in the biosynthetic pathway for the purple color that you see in petunia flowers, and they added extra copies of that biosynthetic gene to their petunias. So what they expected they would get is a flower with a more intense pigment because they had more of the enzyme that makes the pigment. But they didn't get that. What they found was they actually had some completely white flowers and they had these variegated flowers. So adding a copy of the gene was actually suppressing the gene. They also didn't know how that was happening, but they called this co-suppression. And the same phenomenon had been seen and was seen later in other plants and fungi. And I'm going to tell you that that is also an RNAi mechanism. And so RNAi can silence plant genes as well as silencing the viruses. So what did they think was happening? They thought that perhaps the interference was at the level of the RNA, and people started doing experiments with what they call antisense RNA. So normally when a gene is transcribed, the messenger RNA is what we call in the sense strand, and it's a single-stranded nucleotide molecule. If we were to transcribe the other strand, that would be the antisense strand. It would be completely complementary, just like making the two strands of DNA. Now we have two strands of RNA. They do form hydrogen bonds. And when they're stuck together, then the ribosome cannot make it into protein. So translation is blocked. And so if we make an antisense RNA to a particular messenger RNA, we should be able to use that for RNA silencing. And this was what they thought may be happening in the RNA silencing process. So this is just a reminder that ribosome needs a single-stranded template. So if we make it double-stranded, it cannot be translated. We cannot then have, so this is post-transcriptional gene silencing, a block in the translation. So as early as 1994, 1997, people started using this antisense to change genes by adding an antisense to a particular gene, then through genetic modification to change a phenotype. The very first one that was on the market in 1994 in America was the Flavor Saver tomato. They had an antisense to the polygalacturonidase gene, and that means that the cell wall doesn't soften so easily. So the fruit didn't spoil as quickly and had a longer shelf life. Didn't make any change to the flavor, but they called it Flavor Saver, which was a bit misleading. It was actually a shelf life saver. And antisense to the coprotein gene of the papaya ring spot virus was used. And if we have antisense to the virus gene, then the virus couldn't complete its life cycle. This is a plant that is not protected and it's completely damaged by the virus. This was one expressing the antisense to the virus gene, and it was protected against the virus way back in 1997. And this is one of the ways that the Hawaii saved its papaya industry with these new GM plants that were virus resistant. So antisense RNA seems to work, but it was found that people had misunderstood the mechanism. And it was found much later. Remember, those were back in the 1990s. And then in 2006, the award went to Nobel Prize for Physiology and Medicine went to Faramello for their discovery that it's actually double-stranded RNA that is causing the regulation in a sequence-specific manner, or what we call RNA interference. So actually, their experiment was using antisense, but one of their control experiments, they included double-stranded RNA. And what they found was that double-stranded RNA caused a stronger silencing than antisense. And they could have just decided that they made a mistake or the answer was wrong. But instead, they decided to find out why and look at the double-stranded RNA. And they found that if double-stranded RNA is seen inside the cells quickly chop it into small pieces. And those small pieces group together with a number of other proteins known as the RNA inhibiting silencing complex. And that brings one strand of the RNA to its complementary messenger RNA and targets it for degradation. So sequence specific, based on the double-stranded RNA, will target a messenger RNA that is complementary and prevent it from being translated. This was discovered in C. elegans, a worm that is used as a model in genetics, and that won them the Nobel Prize because of physiology and medicine. There is no Nobel Prize for plants. But actually, a year earlier, David Bolcom's group had shown that it is a cytoplasmic RNA trigger that is responsible for virus resistance and homology-dependent gene silencing in plants. So the same phenomenon had been shown a year earlier in plants, but unfortunately, there's no Nobel Prize in plant biology. But don't worry, David Bolcom was given a knighthood, and he, because he had discovered it first, actually was the first to patent this mechanism. So it soon became clear, and this is a review from 2006, that double-stranded RNAs are actually active in eukaryotic cells, long known to be active in bacteria, but now seen to be active in eukaryotic cells as a natural mechanism for controlling gene expression. So in the cell, certain genes, now known as microRNAs, which fold into double-stranded RNAs, are naturally used to control gene expression. Also, viruses are infecting, and their double-stranded RNA triggers gene silencing to attack the virus. All of these precursors are double-stranded RNAs. They trigger the reaction. They're cut into small siRNAs, short interfering RNAs, that can trigger these three mechanisms of gene silencing, degradation, and inhibition of translation. So once it was known a little bit better how these mechanisms worked, people could start using them in biotechnology. We can make artificial microRNA structures, so a single-stranded RNA that bends into a double-stranded RNA to trigger silencing of a particular RNA, or we can use, or we can look at it as a natural mechanism that protects against certain phenotypes. So I mentioned the tomato and the papaya, and later on we had virus-resistant potatoes, capsicum, tomatoes, beans, up till 2011. Then people started targeting not just viruses, but other genes, like a potato with low acrylamide content, a non-browning apple that came into the market in the United States and North America in 2016, and some more that are in development: enhanced nutrient content, more antioxidant levels in tomatoes, reduction of caffeine in coffee, reduction of the allergenicity proteins in peanuts, and even a tearless onion. They reduce the expression of the lacrimatory factor synthesis. So Some of these were antisense, the early ones, and later on they actually used double-stranded RNA because it is a better trigger to form all of these genetically modified plants with changed traits. All of these are genetically modified. That means they have to add new genes into the plants to make the antisense or double-stranded RNA. But not everybody is very keen on genetically modified organisms. And they're expensive and take a long time to develop. If we want to make a genetically modified plant, we need to have a transformation and especially a regeneration system. Maybe we need to be able to make somatic embryos and regenerate the plant. And for some plants, that is very difficult. Also, there is the public perception and concern over eating genetically modified crops. So what if, instead of engineering the plants to make these silencing molecules, we could trigger RNA silencing by just applying the double-stranded RNA onto the leaves? So we don't want to add new genes. We actually only need the double-stranded RNA to trigger gene silencing. Let's just put the RNA on the plant. So we'll call it exogenous RNA to trigger gene silencing. Is it possible? And it turns out it is. This was the first report of that in 2003 by a group in Spain. And here what they did was they have a plant here, and they've infected actually with virus. And what they did was they expressed double-stranded RNA in a bacteria, and then they used that to coat the leaves. The double-stranded RNA was against the virus that they're going to use to coat the leaves. This is the extract from the bacteria that doesn't have the double-stranded RNA, and when they infect with virus, they get symptoms. The same leaf, but now treated with the double-stranded RNA from the bacteria with the double-stranded RNA against the virus, and it protected against the symptoms. So this was the first demonstration that we didn't have to genetically modify the plant. We could just put the double-stranded RNA on the leaf and protect it from the virus. Exogenous double-stranded RNA can protect plants against viruses. This was a very important discovery because we now can cut the time and the cost, and we don't have to genetically modify the plant. So this is just a slide that summarizes using RNAi in agriculture. We can have resistance to viruses, we can prevent spoilage, we can change nutritional value to improve them or reduce specific compounds such as caffeine. We can provide direct protection from pests. We can prolong the efficiency of pest control methods by limiting pest resistance, and we can help prevent infestations by targeting pest development. So these are particularly useful for crop protection, but as I will show you, that's not the only thing you can do with the RNAi. Now, a lot of people might be concerned, if we put the RNA onto the plants, is it actually really not going to change them genetically? Just like many people were concerned about taking RNA vaccines. Isn't it going to change my DNA? Well, just like the RNA vaccines, when we add the RNA, double-stranded RNA, it actually doesn't enter the nucleus. This is a mechanism that completely occurs in the cytoplasm, so it will not alter the RNA. Although the natural mechanism initiates in the nucleus, the silencing part of it actually happens in the cytoplasm. It's triggered by double-stranded RNA, so we can use two complementary strands like this, or we can use one strand so long as it folds into a hairpin. And this is a post-transcriptional mechanism that occurs in the cytoplasm. So remember, I did say there are other mechanisms that occur in the nucleus, but we are not talking about those because they don't happen when we add exogenous RNA. It can only trigger a post-transcriptional mechanism. So this is very safe. There's no genetic change in the plant, but we do get the protection and silencing of the genes, such as the virus, or we can also target genes inside the plant. So now I'm going to move on to why we decided to work on orchids for this technology. Orchids are beautiful and they grow all over the world in almost every country, but especially in tropical climates, we have the most beautiful orchids. And I've been to India and seen the beautiful orchids there too. There's always a demand for new varieties, but breeding takes a long time. They're quite slow growing, although they can be tissue cultured very well. And that would mean that we can do genetic modification. But because they grow slowly, doing the tissue culture and genetic modification is lengthy and expensive. And anyway, we still have the regulatory hurdles for GMO. So we wanted to see if we could use a faster, less expensive method to study gene silencing and for biotech application by using the directly double-stranded RNA. So orchids are highly suitable for double-stranded RNA technology because breeding is slow, because genetic modification is difficult, and we decided to try and target a common virus of orchids, Cymbidium mosaic virus. And it's a good target because it's transmitted by contact. There's no vector. So there's only a short window when the orchid might get infected. It's not infected by insects flying in. It's only likely to get infected when people are handling the plants. So we could actually treat the plants at the time of handling to reduce the virus infection. And we thought there may be also the potential to use endogenous genes for altering flower phenotypes. So what was the potential? We can use silencing to remove or reduce specific proteins in plants, including those expressed in viruses. And we need short double-stranded RNA of the specific gene. We didn't want to do transformation, so we wanted to do the double-stranded directly. So let me tell you a little bit about the virus first. Cymbidium mosaic virus is a double-stranded RNA virus. It's found in almost every country and affects most of the orchid genera in Asia. The infected plants develop mosaics and necrosis, and importantly, it also destroys the flowers. There isn't any treatment or cure, so once you have infected plants, you need to destroy them, and that causes a great loss of income. And although it's not transmitted by insects, it's easily spread on the hands or cutting instruments when you're pruning or when you're handling the plants. Most of the plants start their life free from virus because they're propagated by tissue culture. But once people start handling them, that is the time that they could get infected, and it would be good to protect them against the virus. So here was our strategy. We will make double-stranded RNA using the sequence of the virus coat protein that will be put onto the leaves of the orchid plant. Now, the natural mechanism of the RNA silencing in the plant will be to cut that double-stranded RNA into short sequences, which are complementary to the coat protein gene of the virus. So this is the virus. It is this is TMV, but Cymbidium mosaic virus is in the same group. It's a coat protein with the RNA in the middle. Those coat proteins need to be made by replication in the host. The RNA for the coat protein will be silenced by our double-stranded RNA product, and no new coat protein can form. No new coat protein, no new virus particles, and we should prevent the spread of the infection, even if the plant has become infected. So, how are we going to make our double-stranded RNA? We decided to follow what the Spanish group had done and make it in a bacteria, E. coli. That's a fast and easy and inexpensive way to make our RNA. We cloned a 250 base pair region of the coat protein gene, and we put it into bacterial expression vectors. We put one in the sense orientation under a T7 promoter and one in another vector under the antisense. Both of them produce a 250 base pair band. We then grew our strain of E. coli, and it is an RNase 3 deficient strain so that we can get high yields of RNA. We can induce the production of the RNA from the T7 promoter with IPTG. Then we collect the bacterial cells and lyse them to make a crude RNA extract, which contains the coat protein to the Cymbidium mosaic virus. The extracts are mixed with Cymbidium mosaic virus and rubbed onto the leaves. So we did one set where we used the sense strands of RNA, and one set where we used the antisense strand, and one where we mixed those together to make double-stranded RNA. Then we obtained some tissue cultured plants. We needed quite large numbers for this, so we bought those from a nursery, and it just so happened that they had enough plants of a particular cattleya. So that's why we chose to use cattleya for these experiments, just whatever people had, because we know that this virus infects all the types of orchid. Can you just excuse me 1 minute? I'll try to get rid of the background noise. Sorry about that. I'm over the top of my kitchen. There's a lot of noise down there. I'm trying to get that quietened. Okay, so we used 6 plants in each group, and then we did these treatments, and then we used quantitative PCR to look for the coat protein 30 days after the infection with the virus. And these are the results of the PCR. So in the different groups, I'll explain them. This shows the ones that were infected with virus. This shows the ones that are treated with RNA. So we had various groups. In the first group, we infected with virus, but we didn't use any RNA. So this actually had the highest level of virus coat protein, meaning that the virus had infected the plants. In the next group, we had infected with virus, and we used the sense strand. That actually reduced the level of virus because we get less coat protein. The next group had the virus plus the antisense strand, and that was even better at reducing. But when we used the double-stranded, we reduced the virus level totally to the same as those groups which were not infected with virus at all. One was without any RNA, and one was without virus but with the RNA. And they all effectively had no detectable coat protein in these groups. So that showed us that double-stranded RNA could protect the orchid plants against the Cymbidium mosaic virus. And so, of course, we have to publish that. And we managed to also get a patent, a Malaysian patent, on the use of this technology to protect orchids from viruses. So that was a very encouraging result, and the work of one of my master students. And we were very happy with that result. So we then went on to think, could we actually silence genes of the orchids themselves? Can we use it maybe to alter the flowers? And if so, what sort of genes would be good targets? So our aim was to generate novel flowers through RNA silencing. And we wanted to look at what would be a suitable gene family. And we chose the MIB family transcription factors because these are regulators of anthocyanin biosynthesis and flavonoid biosynthesis, which influence the color of flowers. They also regulate cell shape and maybe the shape of the flowers. And so we wanted to know which of the transcription factors could be useful in flowering. And in this case, we were using dendrobium, and that's because 1, we could get dendrobium plants quite easily, and 2, because we had genome sequence, so we could look up all the gene sequences. So we didn't do the genome sequencing, I should say. That is a published genome sequence for Dendrobium catenatum. We also aligned all the MIB sequences from there, where there are 63 of them that we could find, with those from Arabidopsis and from other plant species. And then we put them into groups by phylogeny. And we looked at which groups had members of MIB family that have been reported in other plants to be involved in flower shape or color. So, for example, the mixture group are involved in epidermal cell shape. The flavonoid group here, and another pigmentation group here. So we selected some of these genes from these different groups to do the double-stranded RNA and test them for their effect on the flower phenotype in the Dendrobium orchid. The first thing we had to do was classify what a normal shape and size of development is. So we did this together with Dr. Trudy Schwarzacker from University of Leicester, and we categorized them into a number of stages and had all the measurements of what the cells really looked like. So then We wanted to identify, or we chose several genes that we thought had a good chance to be involved in flower development. We are then going to make double-stranded RNA against those genes, treat the orchids on the flower bud as the bud is developing. We hope that during this process, the double-stranded RNA will bind to the target transcription factor, reduce the level of transcription factor, which will have an effect on the flower phenotype, which we will be able to observe by looking at the flowers. So the first thing we had to do was to make sure that the genes that we chose actually are expressed in flowers. If they're not, then we won't see any difference if we silence them. So the first one we did, we called DHMIB1 just because it was the first one. And we found that that is expressed during flower development, peaking just before the flowers open. So this is, again, using RT-PCR to determine the level of expression of the gene. So this flower is not treated. This is just what normally happens when the flower develops. We then cloned that sequence into the T7 vector. This time, we didn't do the sense and antisense separately. We just put it between two T7 promoters so that both strands would be made. So we would straight away have double-stranded RNA made from this vector. We put that into the RNase deficient E. coli, express the double-stranded RNA, and then use it to treat the flowers, comparing that to flowers treated with the empty vector without the MIB in as a control to see if there's any difference. Then we observe the flowers during development, especially at the fully open stage. So this was the in the blue, and on the left in each case is the control that is not treated with the MIB, and on the right and in the red is the one treated with the MIB for the buds at stage 5, 6, and 7. So what we can see is there's much less of the RNA in those treated with the MIB, and this is a PCR specific for the MIB gene. So the treatment with the RNA did reduce the level of MIB expression. But if we actually look at the images of the bud, we couldn't see any differences in color or shape. Now, we knew that this particular MIB maybe is affecting cells' shape, but maybe not affecting the color. And it wasn't affecting the flower shape either that we could see. So in order to look at cell shape, then we needed to do some electron microscopy, and this is SEM of the different areas of the petals, the sepals, and the lip of the flower. We also looked at the relative expression of the MIB in the petals, the sepals, and the lip. And what we found was the most reduction was actually in the lip. So when we looked actually at the lip structure cells under the electron microscope, we did manage to see a difference. So this is the shape of the cells treated with double-stranded from empty vector. So we can see that the cells are quite conical and pointy, but on the flowers that were treated with the MIB double-stranded RNA, they become much flatter. If we look at the side, again, we can see pointy and much more dome-shaped. So what we were able to show was some difference at the level of the cells through the treatment with double-stranded RNA, but not a difference that we could see with the naked eye. So it was a good result, but not as exciting as we had hoped. So perhaps we haven't chosen the right MIB gene. So we got another grant, more funding. We went back to look at some more MIB genes. And this time we also included the chocon synthase as a control. If you remember back from my slide on the petunia, which was done by another research group, chocon synthase was the purple pigment color, and we are working with a purple flower in the Dendrobium hybrid. So we cloned and expressed double-stranded RNA for the chocon synthase, the MIB3 and the MIB4, and we also have an empty vector group. We're going to make the double-stranded RNA for each of these and treat plants with either this one, or with this one, or with this one, or with this one, and then compare them across the stages of development. So these were the results at the different stages, where 6 is the open flower and 1 is one of these tiny immature buds. The top row is for treatment with MIB3. The next row, they were treated with MIB4. The next row was treated with chocon synthase. and the last row was treated with empty vector. Now, I hope you can see there is some difference in color. The untreated flower is a more intense purple. If we silence the purple color flower gene, we get a paler flower, and also with the MIB3 and the MIB4, we get a paler flower. We also notice that the shapes of the buds and the petals and sepals were slightly different with MIB3 and MIB4. But how do you quantify the color and the shape? So for quantifying the color, we use a colorimeter and color hexa analysis. So we take a panel of the color, we digitally analyze it for light, dark, red, green, yellow, blue to get a color score for each of these. And then we can compare them to the empty vector. So these were the color scores, and this is the difference from the empty vector. So we actually then get a numerical value that we can plot on a graph. So the color change, so the bigger the bar, the bigger the difference between the untreated and the treated. So the largest changes were in DH-MIB3 treated petals and DH-MIB4 treated sepals, but all of them showed a difference from the control that we could measure based on color analysis. So the color was reduced, and how about the anthocyanin content? So we also measured the anthocyanin content with biochemical assays, and we also found, so here the leftmost bar in each set is the untreated control, so it has the highest level. So all of the treatments with either chalco synthase, DH-MIB3 or DH-MIB4 reduce the amount of anthocyanin at stage 3, stage 4, and in the open flower, petals, sepals, and lip. So we have reduced the color and the anthocyanin content through the use of the double-stranded RNA. What about the shape? This was a little bit more difficult. We're learning things as we go along. So it turns out that you can flatten the flower and then get the image and use graphing software to plot the shapes. So we did for the sepals, we did for the petals, and we also did for the lips. And then we can subtract the difference between the treated and the untreated. So here's untreated shape. This is treated with MIB4, for example. We put the one plot on top of the other, and it has a software that can say how big is the difference in shape. We give it a shape distance measurement. So the taller the bar here, the bigger the change in shape. So you can see we got the biggest change in shape in the petals of the open flower with the MIB3 and with the MIB4. We did also see some very slight changes with the chalcone synthase. So what does all that mean? What we can conclude from our study is that direct application of double-stranded RNA protects orchids against Cymbidium mosaic virus. It's very useful for crop protection against viruses, and now people are starting to use it for other pests and diseases. People are using double-stranded RNA against fungi and against insects as well, which can be sprayed onto the plant. They target, for example, the insect gut, so that when the insect eats the plant, it takes up the double-stranded RNA, and that interferes with it and can actually also control insects. So it's very exciting that all these new products will be able to come into the market soon. They're non-GMO and they're transient, and they can protect crops very well. What else is interesting, though, apart from crop protection, is we can actually change phenotypes such as flowers. We can use this then in studying gene function and potentially to actually modify the flowers. So for our study at the moment is pretty academic. We can maybe say that this is the role in gene function, but eventually if you choose the right genes, you may be able to use it to modify flowers in a particular way that you desire. It's much more rapid and inexpensive than doing genetic transformation. So even if we're not talking about a commercial product where we're worried about genetic transformation acceptance, it's actually better when it's cheaper to use for functional studies in the lab too. So it can be a useful research tool. And we are continuing to test new candidates for flower modification and trying to finish the paper on the flower color that I've just shown you. What about the prospects for commercial use of double-stranded RNA in agriculture? So RNA sprays have already been demonstrated as effective pesticides. And one of the main concerns that people had is that RNA may be not a very stable molecule. So if it's going to break down quickly, could that be actually a problem for in the field use? Do you have to spray it every day or 2 times a day? Won't that become more expensive? So actually, people have found that by using carriers such as clay nanosheets, you can improve the stability of the RNA. It can last for several days, and that has been actually shown for tomato viruses in Australia. What about the potential for use in floriculture? We think so, and we are continuing to work on that direction. It's perceived that this may be more acceptable to the public because there's no issues of GMO, and that maybe will make it easier to commercialize as well. However, the current biosafety guidelines aren't always clear on this type of technology. Is it a GMO or not? If we made it in a GMO bacteria, does that mean the bacterial extract is a GMO, or is it a pesticide? So it's interesting that countries are starting to consider this. So Australia recently has exempted topically applied double-stranded RNA from a GMO regulation. So if you make your RNA and you just use the RNA on the plant, it is not regulated as a GMO, but it is regulated as an agrochemical. And New Zealand has also gone the same way. And we think that countries like Malaysia and India, we both have very similar biosafety regulations in Malaysia and India, are quite likely to also go for this exemption. That means we will regulate it, but not as a GMO, but as a pesticide or as some kind of agrochemical. So it's been in the news, gene silencing sprays, first with the tomato paper that came out in 2017, there was a lot of excitement, but just this year, apart from crop protection, tunable crops are just a spray away. Reprogramming for agronomic performance. So here in 2021, reports saying that we can alter the phenotypes of plants as well and not just use it for crop protection. So it's quite an exciting time. So before I finish, I again thank you very much for the invitation and for hosting this seminar. I have to thank my funding agencies, which include the Ministry of Education, Malaysia, for these various grants that we've had. This was the product of several different research projects and several different researchers. So it's a lot of work, and particularly I have to thank the students, Teng Wei, Su Yi, and Ashraf for this work. Also my collaborators, Dr. Purabi Mazumdar, Prof. Yasmin Othman, and Dr. Trudy Shosaka at University of Leicester. This is Purabi, and this is Su Yi. I don't think we have Teng Wei or Ashraf. I need to get a new picture. Thank you very much, and I'm very happy to take questions.
Thank you, ma'am.
The first step to receiving knowledge is being brave enough to ask a question. So now the session is open to queries. Ma'am, we have a question in the chat box. Is RNA silencing used in any other ornamental plants other than orchids?
Okay, thank you for the question. Actually, I don't think it has yet been commercially used, but in research, yes. So we mentioned the petunia. Another flower that is another flower that is often used in research is the what we call the snapdragon or the antirrhinum. It has been used, but not yet commercially. Yeah, it should, in theory, be able to be used in any types of flowers. Yes.
Ma'am, can you hear me, ma'am?
Yes.
Ma'am, I have a question. Your session was very clear and audible, and we had a good interaction. Ma'am, I have a doubt, ma'am. Yes. Ma'am, new gene candidates we can do in some other flowers apart from orchids?
Yes, yes, I think so. The the only issue is that you need to know the sequence of the gene that you want to target. Yeah, because it's based on the sequence. So if you want
She's
to
answering.
silence a gene, you need to have a good or a quite accurate gene sequence to make your double-stranded RNA. If it is
Yes.
too different, it may not target the RNA that you want to silence.
Yes.
So there's two ways you can do that. When we first started with the MIG1, we actually just cloned that gene and sequenced it ourselves based on similarity to other species. But for the other ones, it was a bit easier because the dendrobium genome sequence had been published. So we were able to do that. So the same thing for the viruses as well. For most of the viruses, it's quite easy to clone and sequence the genes yourself. That's not that expensive. So I think that's why a lot more work has been done on viruses so far.
Thank you, ma'am. Thank you.
So
Ma'am, I have a question, ma'am.
sorry, I just have one, just to ask Dr. Caroline again. What flowers would you be interested in?
No, ma'am. We thought we'll try. Apart from orchid, we have local flowers, many flowers. So we
Yes,
asked. Is
sure.
flowers name, ma'am, like
Right,
hibiscus,
right, yeah.
and asclepias. Any asclepiaceae family members,
Mm.
carnations, and anthuriums. We can do, ma'am.
Yeah. So I
Yes.
think for some of those species, you may be able to find that the sequences are available. But even when they're not, if you're using something like the transcription factors, they're quite highly conserved. So you can quite easily clone them to get the sequence. So it's possible. Yeah.
Thank you, ma'am.
Sorry,
Thank you.
I
Yes,
interrupted. Somebody has another question.
yes, ma'am.
Yes, ma'am. Ma'am, my question is, can we treat mutation in plants using this RNA silencing, ma'am?
Can we treat mutation?
Yes, ma'am. The genes are mutated, ma'am, when there is an addition of gene or something like that. Can we use RNA silencing to treat or suppress those added genes, ma'am?
Okay, so we can use it to target any gene that is expressed. So if you think about it, if the mutation is causing expression of something you don't want, so this is how it may be used medically. For example, there may be some genes expressed in diseases like cancer that you don't want them expressed. So you could then use the RNAi method to try to silence the expression of anything that you don't want expressed that is causing a problem. So in terms of cancer, that is a mutation that you're targeting. So yes, it can be used in that way. Yeah.
Thank you.
Any other queries, students?
Don't be shy. There is no such thing as a silly question because this is all quite a new area. A lot of things are not known yet. So you may have what you think is a silly question is not a silly question. Please do ask. Don't be shy. I think maybe everyone is tired. You've had a busy day, yeah?
If the queries are over,
If the queries are over, we can move on, ma'am. Thank you for your valuable answer, ma'am.
Thank you.
gratitude is a powerful catalyst for happiness. It's the spark that lights a fire of joy in your soul. I request Dr. U. Danya, Assistant Professor, Department of Botany, to express her gratitude.
Challenges and problems are the fuel of success. Every experience in your life being orchestrated to teach you something you need to know to move forward. Good afternoon, one and all present here. On behalf of Botany Department, I extend a hearty vote of thanks to our resource person, Dr. Jennifer Ann Harikrishna, Genetics and Molecular Biology, Institute of Biological Sciences, Faculty of Science, University of Malaya, Malaysia, for being with us and accepting our invitation in her busy schedule. Ma'am clearly explained about RNA interference. Post-transcriptional gene silencing is a conserved biological response to double-stranded RNA that mediates resistance to both the endogenous parasites and the exogenous pathogenics and the nucleic acids and regulate the expression of protein coding genes. And then RNA interference in the field of biotechnology, which having managing the pest disease caused by the bacteria, fungi, viruses, and then improving crops, and then generating the plants with the novel traits. Thank you very much, ma'am, for your excellent and informative, elaborative lecture. And the photos are very helpful to understand the concept, ma'am. Thank you very much. I especially acknowledge our Secretary, Reverend Dr. Sister Ruby Alangara Mary, our Principal, Reverend Dr. Sister Helen, our Vice Principal, Reverend Dr. Sister Mary Fabiola, Controller of Examination, Reverend Dr. Sister Emelda Mary, Dean of Academics, Mrs. Polencia Kamal, Advocacy Coordinator, Dr. Pauline Vasanthi Joseph, and Dean of Students Affairs, Dr. U. Mary, for their constant support and encouragement. I would like to thank Dr. Carolyn Jo Rosario, Head of the Department, who is the source of inspiration and motivation. Thank you, ma'am. This vote of thanks would be largely incomplete without expressing my gratitude to my dear colleagues who have been working really hard and coming out all the allotted duties very meticulously. Last but not least, I thank you, my beloved students and scholars, for their patient listening. Thank you, one and all. Thank you.
Thank you, ma'am. A good plan today is better than a perfect plan tomorrow. Don't wait for an inspirational ending to come to mind. Work your way to the ending and see what comes up. Thank you all.
Thank you, ma'am, for sparing your time with us.
Thank you for the invitation. It's
It's
very nice to see you all again.
in future, ma'am, we'll have